Dear students, to understand the role of oxidative stress in the pathogenesis of conditions such as cancer, complications of diabetes mellitus, and atherosclerosis, we must understand the term oxidative stress. And to understand the oxidative stress, we must know what are the free radicals and what are the antioxidants. So I'll begin with free radicals. Free radical has an unpaired electron in its outermost shell. In a quest to fill their partially empty valence shells, free radicals run around the body, stealing electrons from other atoms in our cells and tissues. This electron stealing oxidatively damages cells, proteins, DNA, RNA, and lipids. And it is an underlying factor in many chronic diseases, including cardiovascular diseases, type 2 diabetes mellitus, cancer, and various autoimmune diseases. Free radicals are highly reactive, short-lived species which the lifespan may vary from 10 to the power minus 9 to 10 to the power minus 12 seconds with an unpaired electron. Upon collision of a free radical with other molecules, they can either abstract or donate an electron in order to gain stability. This leads to the generation of radical form of the molecule with which the free radicals have collided, causing a chain reaction. Free radicals are generally represented by superscripted dot, as you can see here. There are numerous reactive species such as superoxide anion radical. It is a reactive oxygen species as well as an anion. It has a half-life of about 10 microseconds at 37 degrees. Hydroperoxyl radical. It is a reactive oxygen species. It is the protonated form of superoxide anion. Hydrogen peroxide is not a free radical because it has no unpaired electron in its outermost shell. But it is able to form highly reactive hydroxyl radical through Fenton or Hebowit's reaction. Hydroxyl radical, it is a highly reactive oxygen species. It has a half-life of only 1 nanoseconds at 37 degrees. Lipid peroxide radical, it's a secondary radical. It has a half-life of about 10 milliseconds. Nitric oxide is produced by the action of oxygen on the amino acid arginine in the presence of the enzyme inducible nitric oxide synthase. And this is a reactive nitrogen species, and it has a half-life of about few seconds at 37 degrees. Peroxynitride is a reactive anion, and it is synthesized by the interaction of nitric oxide with superoxide, which results in the formation of peroxynitride. Peroxynitride, though it's not a radical species, it has no unpaired electron, but it is included in this category because of its highly reactive nature. Some important characteristics of reactive nitrogen and reactive oxygen or reactive nitrogen species include their extreme reactivity. They have a very short lifespan, and most of them act where they are produced. They are involved in the generation of new reactive oxygen species through chain reaction. They damages various molecules, thus causing the damage to the cells and to the tissues. Free radicals can be generated in healthy humans. The major source of free radical in cells is the electron leakage from the electron transport chain. Normally, efficiency of electron transfer is so high that most of the oxygen is completely reduced to form water, and only less than 1% of the oxygen forms reactive oxygen species. Transition metals which can amplify the free radical response or which can amplify the generation of free radicals are normally kept sequestered. However, some electron leakage is inevitable under normal circumstances. Superoxide radical, this is the superoxide radical, is produced when a single electron is transferred to oxygen. As I have already mentioned, superoxide is an anion as well as a free radical. Hydrogen peroxide is formed by the 2 electron reduction of oxygen. Since reaction generates a non-radical species from the radical reactants, it is called as a dismutation reaction. Dismutation can occur spontaneously, as you can see here, or it can occur or it can be catalyzed by superoxide dismutase. The 3 electron reduction product of oxygen is hydroxyl radical, which is the most powerful free radical. Hydroxyl radical is also produced when an electron is transferred from a ferrous ion to hydrogen peroxide. Fe2+ can participate in Fenton reaction, generating highly reactive hydroxyl radical. Another iron catalyzed reaction for the generation of hydroxyl radical is called the Haber-Weiss reaction. Superoxide anion participate in the Haber-Weiss reaction, which combines Fenton reaction and the reduction of Fe3+ by superoxide, yielding Fe2+ and oxygen. It is important to know that low quantities of reactive oxygen species are formed during normal metabolism. Auto-oxidation of thiol, ascorbic acid and glucose are greatly amplified through participation of transition metal, and this causes generation of reactive oxygen species. Flavine coenzymes present in the peroxisomes are quite active in generating hydrogen peroxide. FADH2 and FMNDH2 get oxidized when they transfer their hydrogen directly to molecular oxygen, resulting in the generation or the formation of hydrogen peroxide. Enzyme xanthine oxidase catalyzes the reaction, which results in the generation of reactive oxygen species. The reaction catalyzed by this enzyme result in the formation or conversion of hypoxanthine to xanthine with the formation of hydrogen peroxide. Similarly, xanthine oxidase causes the conversion of xanthine to uric acid with the concomitant generation of hydrogen peroxide. The cellular consumption of oxygen is greatly enhanced by both the cell types, that is the phagocytic cells, and the bacteria after the ingestion of bacteria by phagocytic cells. In neutrophils, the oxygen consumption may rise up to 50-fold. The process is known as respiratory burst. Respiratory burst involves the activation of membrane-bound NADPH oxidase located on the phagocytic surface. The enzyme NADPH oxidase catalyzes the formation of superoxide anion. Dismutation of this superoxide anion results in the generation of hydrogen peroxide. Hydroxyl radical can be generated from this hydroxyl radical can be generated from the superoxide anion or from hydrogen peroxide by Haber-Weiss or Fenton reaction. Hydrogen peroxide is also involved in the generation of another lethal reactive oxygen species, that is hypochlorous acid. The enzyme catalyzing the reaction, this conversion or this reaction is myeloperoxidase, which is present in the neutrophils. Hypochlorous acid, superoxide anion, and hydroxyl radicals are involved in the killing of bacteria. This is the deliberate production of free radical by the body for killing the bacteria. I have already mentioned that how ferrous iron is involved in the regeneration of reactive oxygen species. Like the ferrous iron, copper in the cuprous state can also react with hydrogen peroxide to yield the hydroxyl radical. The ferrous iron and the cuprous copper are more reactive than their oxidized counterparts. In their absence, superoxide and hydrogen peroxide are harmless and rapidly removed from the body. Therefore, they do not exist in free form normally in the body. In plasma, iron is bound to apoferritin to form transferrin to form transferrin. While being stored, iron is bound to apoferritin to form ferritin. Serum copper is transported bound to ceruloplasmin. There are numerous exogenous sources of reactive oxygen species. Various compounds such as carbon tetrachloride is capable of generating oxidative stress. Ionizing radiations damages tissues by producing free radicals. Light of certain wavelengths can cause photolysis of covalent bonds to produce free radicals. Cigarette smoke also contain dangerously high concentrations or high levels of free radicals. Other exogenous sources of reactive oxygen species include polluted air, water, alcohol, heavy metals, transition metals, drugs such as cyclosporine, tacrolimus, gentamicin, industrial solvents, cooked items, and cooking medium. For example, smoked meat, repeatedly used oil and fat. ROS, though more commonly known for their harmful effects, but ROS also plays several important roles in number of biological processes and regulates cell physiology and functions. reactive oxygen species become dangerous to the human body when they produces oxidative stress. Oxidative stress arises from an imbalance between oxidants or reactive oxygen species and antioxidants. And this imbalance is in favor of excessive generation of free radicals or reactive oxygen species. Increased oxidative stress causes the oxidation of biomolecules with consequent loss of their biological functions. Build up of reactive oxygen species, whether they are endogenous or exogenous, may result in lipid peroxidation, protein oxidation, and DNA damage, to name a few. Nucleic acids, DNA and RNA, are highly prone to hydrolysis, non-enzymatic methylation, oxidation, etc. DNA can be damaged by reactive oxygen species in two ways. ROS can modify base or sugar. If ROS modifies a base, it can add hydroxyl group to the base, and the hydroxylated bases are different from the normal bases. Oxidative damage to DNA results in several mutagenic lesions. The most susceptible base in DNA that undergoes oxidation most radially is 2-deoxyguanosine, resulting in the formation of 8-oxo-2-deoxyguanosine. This is an oxidized derivative of 2-deoxyguanosine. The concentration of 8-oxo-2-deoxyguanosine within a cell is measured to determine the level of oxidative stress. The most prevalent base in RNA which undergoes oxidation is guanosine, resulting in the formation of 8-hydroxyguanosine. Modification of sugar causes the strand breaks. DNA can be modified in variety of ways. If it is not repaired, can ultimately lead to mutations and genomic instability. This could result in the development of variety of cancers, including colon, breast, and prostate. Several oxidative changes in proteins have been described. The self-hydryl group of cysteine is highly prone to oxidation and can result in the formation of disulfide bridge. If reactive oxygen species is strong enough, that means it is more aggressive, then it can oxidize cysteine side chain, that is the self-hydryl group, to various acids derived from sulfuric acid, such as sulfinic, or sulfonic acid. And once the cysteine is oxidized in this form, it cannot be repaired. Protein carbonyl is formed by the oxidation of side chain of amino acid lysine, arginine, proline, and threonine, residues of protein bound or free amino acids. Protein carbonylation refers to a process that forms reactive aldehydes or ketones. Also, the epsilon amino group of lysine and guanidino group of arginine can react with carbonyl groups of carbohydrates, resulting in the formation of advanced glycation end product, which is also known as AGs. Oxidation of protein can cause impaired protein folding, backbone fragmentation, resulting in loss of function. These oxidatively modified proteins are usually recognized and degraded in the cell, in cells, but some of them can accumulate over time and can lead to cellular dysfunction. Low density lipoproteins are the major carrier of cholesterol to body tissues. The oxidation of LDL is a complex process during which both the protein as well as the lipid component of the LDL can undergo oxidative changes that can cause cholesterol accumulation. Polyunsaturated fatty acids, in particular linoleic and arachidonic acids, are other important targets of lipid peroxidation, as it proceeds in a self-amplifying manner, which is referred to as chain reaction. See, oxidation is a loss of electron. However, we call oxidation of lipid as lipid peroxidation. This is also a loss of electrons, in which lipids are in loss of electrons in lipids, and the intermediate generated is called peroxyl radical, and therefore we call this as a lipid peroxidation reaction rather than lipid oxidation. Lipid peroxidation is a three-step process, that is initiation, that is the attack of a free radical on the lipid molecule, and the propagation reaction, in which a fatty acid radical react with the oxygen, and the third step, which is the termination step, in which the two reactants, they collide with each other to form a non-radical species. So the initiation phase starts with the attack of free radical on one of the methylene hydrogen. This hydrogen, the hydrogen between the two double bond, is highly reactive. This wants to become water, so it's going to take the hydrogen from the fatty acid or the lipid molecule, and this is going to be converted into water molecule, and the lipid molecule will be converted into its radical form. So the chain reaction is initiated with the interaction of polyunsaturated fatty acid with free radical. The removal of the hydrogen generate the radical form of the lipid, which is, and this radical form of the lipid is carbon centered radical, and this is called as the reaction 1 or the initiation phase. This initiation phase of lipid peroxidation can be better visualized in this slide. The next is the propagation phase. This promptly react with oxygen to form the fatty acid peroxyl radical. Oxygen molecule react with the lipid radical generated in the initiation step to form fatty acid peroxyl radical, which you can visualize in a better way in this slide. This reaction is termed the carrier of the chain reactions, since it can attack another polyunsaturated lipid molecule. This can now react with another fatty acid, or it can and then it can strip a hydrogen atom from another fatty acid, resulting in the generation of lipid hydroperoxide. This can be better visualized in this slide. This is the lipid peroxyl radical. It reacts with another unsaturated fatty acid. It strips a hydrogen from the unsaturated fatty acid, resulting in a generation of lipid peroxide as well as a lipid radical. Like hydrogen peroxide, lipid hydroperoxide can also react with iron or copper and form peroxyl radical. Damage caused by these species is not confined to their site of production, since they can diffuse to other parts of the cell. Termination of this reaction chain occurs when a radical reacts with another such same or the different radical to form the inactive products. For example, the reaction of these two can generate the non-radical species. Similar radicals or different radicals can react to terminate the reaction by the generation of non-radical species. Lipid peroxidation disrupts the normal structure and function of lipid bilayers surrounding both the cell itself and in the membranes of organelles. In particular, the lipid peroxidation can alter membrane permeability, transportation, and fluidity. With this, I have finished the free radical. Now we will start discussing the antioxidant defense systems of the body. Antioxidants are substances that neutralize or remove free radicals by donating an electron. The neutralizing effect of antioxidants help protect the body from damage caused due to oxidative stress. An antioxidant is any substance that, when present in low concentration as compared to those of an oxidizable substrate, significantly delay or prevents the oxidation of that substrate. Antioxidants are the substances that help protect the body from free radicals. Anti means against, and in this case, against oxidants. And oxidants, when present in higher concentration, are harmful to the body. Antioxidants can be water soluble, such as ascorbate, uric acid, polyphenols, glutathione, cysteine, transferrin, and many more. Lipid soluble include alpha tocopherol, carotenoid, and retinoids. Antioxidants produced by the body include catalase, superoxide dismutase, glutathione, transferrin, ferritin, ceruloplasmin, albumin, uric acid, bilirubin, thioredoxin. And those which we obtain from the diet include beta-carotene, ascorbate, vitamin E, selenium, polyamines, polyphenols, and flavonoids. Antioxidants donate an electron to a free radical and neutralize them. This causes severe reduction in the reactivity of the free radicals. Free radicals are electron thieves and antioxidants are electron donors. What makes antioxidants unique is that they can donate an electron without becoming reactive entity themselves. The unpaired electron in the antioxidants is delocalized and therefore it is not reactive. Antioxidant protects cells and safeguard their constituent from damage caused by highly reactive free radicals. Every antioxidant behaves differently due to different chemical properties. There is no universal antioxidant which can counteract all the different types of free radicals generated in the body. Some antioxidant may become pro-oxidant, such as vitamin C, which can grab electron from other molecules, create chemical instability, and may produce oxidative stress. Although vitamin C is an antioxidant capable of reacting with superoxide and hydroxyl radical to yield monodihydroascorbate and hydrogen peroxide, but it can also be a source of superoxide radical by reaction with oxygen and hydroxyl, by reaction with oxygen and the hydroxyl radical by the reaction with Cu2+. However, these pro-oxidant actions require relatively high concentration of vitamin C, which are unlikely to be reached in tissues. Free radicals, as you know, are unstable, short-lived species with an unpaired electron. Free radicals are produced within the body as byproduct of normal metabolism, natural aging, and as a result of exposure to radiation, stress, and some environmental pollutants. Free radicals may produce different effects in different areas of the body because of the difference in the chemical structure. It can react with virtually anything in its vicinity. Free radicals are highly reactive, and when present in excess, they can damage virtually all cellular components. In healthy individuals, a delicate balance exists between the levels of free radicals and antioxidants. When this balance is disturbed and it is in favor of reactive oxygen species, then it can play an important role in the causation in causing variety of diseases. In some pathological conditions such as diabetes mellitus, and in critically ill patient, oxidative stress increases because free radicals are increased. So what is oxidative stress? Oxidative stress is an imbalance between antioxidant and reactive oxygen species, and this imbalance is in favor of increased production of reactive oxygen species. Body has developed defenses against free radical. To fight free radicals or keep the free radicals under check, body has pool of antioxidants such as enzymatic antioxidants and non-enzymatic antioxidants. Together, these antioxidants ensure mopping up of the free radicals as they are produced. Enzymatic antioxidants include catalase, superoxide dismutase, glutathione peroxidase, and glutathione reductase. Superoxide dismutase has extremely high activity and is widely distributed in all tissues. It catalyzes the removal of superoxide anion. The reaction involves 2 superoxide anion and 2 proton. And as you can see here in this slide, one of the superoxide anion is oxidized to oxygen, the other is reduced to water. Such reactions are called as dismutation reaction. There are 3 isoforms of superoxide dismutase. SOD1 is found in the cytosol, which is zinc and copper containing. SOD2 is found in the mitochondria, which contain manganese. And other SOD, that is SOD3, is found in the extracellular fluid. It also contains zinc and copper. Catalase catalyzes the decomposition of hydrogen peroxide to water and oxygen. It is an active heme protein present in almost every cell, for example, liver, kidney, bone marrow, blood. Phagocytes are rich in catalase. As you can see here in this slide, one of the hydrogen peroxide is reduced to water, the other is oxidized to oxygen. Glutathione oxidase is a selenium-containing enzyme that catalyzes the reduction of hydrogen peroxide and organic or lipid hydroperoxide in using glutathione as a hydrogen donor. Glutathione peroxidase helps in maintaining low concentration of hydrogen peroxide or organic or lipid hydroperoxide in tissues. As you can see in this slide, hydrogen peroxide is converted to water, whereas the glutathione is converted into its oxidized form, and the organic or the lipid hydroperoxide is converted into alcohol derivative. The oxidized glutathione is reduced by the action of glutathione reductase. The non-enzymatic antioxidants, they act by donating a hydrogen to the free radical. The non-enzymatic antioxidants can be obtained from the food, or it can be synthesized in our body. So those which we obtain from the food include these, whereas the one which is non-nutrient is included in this category. Among the nutrient antioxidants, vitamin E and C are the most important. Vitamin C is water soluble, whereas vitamin E is highly lipophilic and is localized in membranes and as part of lipoproteins. Antioxidants such as glutathione peroxidase, catalase, and EDTA, they have preventive role, means they prevent the initial generation of free radicals. They are able to destroy the oxidizing species before the damage can be done. Glutathione peroxidase is found both in cytosol as well as in mitochondria. As we have already discussed, it eliminates hydrogen peroxide, thus preventing the unchecked buildup of peroxides in cell. The oxidized glutathione is reconverted to reduced glutathione by the enzyme glutathione reductase. Detoxification of hydrogen peroxide in this way prevents the generation of free radicals from it. Catalase is another enzyme that causes the decomposition of peroxide to water and oxygen. Catalase is a heme-containing enzyme which is widely distributed in various tissues. There are antioxidants, both enzymatic as well as non-enzymatic, and found both in aqueous and membrane compartment, and their mode of action is interceptive or scavenging. Superoxide dismutase is the only enzyme that takes free radical as a substrate and hence act as a scavenger. Non-enzymatic antioxidants include all this. As I have already mentioned to you, they mostly act by donating hydrogen to free radical, and they themselves become radical species. These radical species are not very reactive because their electrons are delocalized, and generally the non-enzymatic antioxidants they have a very small molecular weight. Vitamin E, beta-carotene, vitamin E, ubiquinone, and beta-carotene retinoids are being lipophilic. They act on biological membrane. Rest are aqueous phase antioxidants. Vitamin E and beta carotene are also called chain-breaking free radical trapping antioxidants since they terminate the lipid peroxidation reaction. As you can see here, they react with the tocopherol. Vitamin E reacts with a free radical, and itself it is converted into tocopheroxyl radical. Tocopherol is the most potent chain breaking antioxidant in tissues. Beta carotene is less effective. Antioxidants are grouped into three classes on the basis of their location, that is intracellular location, those which are found in the extracellular fluid, and the membrane associated antioxidants. So you can go through this table. First line of defense is provided by the antioxidant that act to suppress or prevent the formation of free radicals or the reactive species in cells. They are very fast in neutralizing any molecule with the potential of developing into free radical, or any free radical with the ability to induce the production of other free radicals. The three key enzymes include superoxide dismutase catalase and glutathione peroxidase. This class also include metal ion binding proteins like transferrin, ceruloplasmin, which chelate or sequester iron and copper respectively, consequently preventing them from free radical formation. This group of antioxidants is often referred to as scavenging antioxidants. Second line of defense is provided by most antioxidants, including vitamin C or ascorbic acid, uric acid, glutathione, alpha tocopherol, and ubiquinone. Most antioxidant belongs to this category. Consumption of food rich in antioxidants may reduce the risk of various diseases such as cancer, diabetes mellitus, rheumatoid arthritis, and may slow down the progression of age-related muscular degeneration. Antioxidants are regularly used in food industry to increase the shelf life of products. Antioxidants added to food to prevent the oxidative damage to oils, particularly those containing polyunsaturated fatty acid, and therefore it prevents rancidity. Thank you very much. With this, I have finished the free radicals and antioxidant defense systems of the body.